So for today's talk, I'm going to talk about impacts of updated standards on the IFU for reusable validation and for reusable devices. If you do make reusable devices, how the standards have affected the IFU. Okay. For the agenda, we're going to go over what reusable medical devices are, how they function in the market in terms of standards, the role of the validations that they play, impacts that are required, how the instructions for use have been impacted over time, and what is next for cleaning validations, because that is the most important things that have updated recently. And what do manufacturers need to be ready for? There's a lot of uncertainty out in the industry right now when you talk about reuse validations for devices. And it is that period that we can help shape the industry and regulatory on how to move forward. And they are looking for a partnership with the manufacturers and other organizations to help them define, because we are in that uncertain zone right now. For those of you that don't know reusable devices, these are the devices that are used on different patients, but they have to be reprocessed. They have to be cleaned, disinfected, and sterilized, and then reused over and over again until indefinitely, right? And I will go over at the end of the presentation what the indefinitely means and how are we assessing that. This gives you an overview of what different categories are for reusable medical devices. There's just not critical devices, right? Our understanding usually is these are devices that are invasive going into the body, and there are critical devices, but we're also looking at home care products, right? We've done breast pumps, we've done menstrual cups, we've done diabetes monitoring systems, a lot of different things, CPAPs, right? All those need to be validated. And we are working on products not only for hospital healthcare, but clinics like dental industry and also home care products. And I've got here capital equipment, which I will talk about here. And if you see here, all the orange spots that I've identified in this slideshow is what we have actually validated in-house for cleaning and disinfection. And these are devices that manufacturers make. But the misconception here is that these devices are for practices. Hospital practices will take care of it. There are organizations in healthcare industry that provide procedures to hospitals of how to sanitize and how to disinfect devices. But now we are moving towards, after COVID, it has taken a shift where these kind of devices didn't have, they had hospital practices, but no validation to show that it does work. If I wipe for this many, does it really kill the pathogens that should be killed? And that's when the manufacturers were contacted and said, where's the instructions for use on that, right? Do you even have instructions for use on that? You manufacture it, sell it, and hopefully hospitals taking care of it. But now we're seeing that more and more of these equipment are coming in for validations, and these are non-critical devices that are, some are patient contacting, some are not patient contacting. And even mattress covers we see come in for reprocessing, just wiping. Although you have a cover on it and you take it out, we still have to validate it for cleanliness and disinfection. And I will go over the disinfection in a little bit on the criteria. Now, what determines a standard development? Now, when we write standards, what are we looking for? We're looking for what is happening in manufacturing, what is happening in healthcare facility. As a testing facility, you provide your instructions for use to me. My job is to make sure that I'm validating for the manufacturer, but I'm also incorporating the healthcare industry, the people that are actually reprocessing the devices. My protocols have to make sure that the instruction for use that you have provided is challenged and it mimics what healthcare is doing. Because if you think about it, you are selling a product to healthcare, they're cleaning it, we validated it, and for the longest time, there's been that huge gap between healthcare and testing labs, right? Manufacturers and healthcare, they work together, they train, they figure things out. But when we come into the play, we're validating your IFU. And if we are not in the same realm as they are, then we're missing a huge part. And so we get involved in these industries now. We go to all the standards and the organizations and try to learn what they're doing and incorporating those into your validation plans. So you may be like, yeah, it's not part of our IFU. Why do you have this in there? But that is what we're trying to do, is we're trying to take those simple things and bring it into our protocols to ensure that the validations are happening according to how they're being processed in the hospital. And taking your IFU. So we're taking two things and meshing them together. Now for instructions for use, the major impact is the cleaning, right? Cleaning is the first step in any reprocessing, and if you don't do that correctly, nothing is happening after the fact. You can't disinfect it, you can't sterilize it, it just goes hand in hand. So you want to make sure that you are addressing that portion first. Now, point of use cleaning, every manufacturer will have a point of use cleaning. And what happens with point of use cleaning is we omit that from your instructions for use. And the reasoning for that is not every facility does point of use cleaning right away, right? You do surgery, you put it in a pan, and then that pan goes somewhere in the basement in a hospital, and they're cleaning it. But are they spraying it with a detergent or a foam that's going to keep it moist? Are things happening? And so because of that, we challenge the process that, guess what, they forgot to do pre-processing. And so we're taking it a step further and omitting the step. The only time this step is included is when you are reprocessing flexible endoscopes. That is part of their IFU, and it is a requirement for the hospitals to do that. That's why. Every IFU has a requirement, but as you manufacturers know, hospital has their own world, and if they want to do it, they'll do it. If they don't, they won't. But in here, disinfections, I've put not defined because a lot of times we don't define them. Manufacturers all over the world have to face, if you are global, disinfection is coming your way. And when I get to disinfections, I'll talk about it. Sorry, I got a thing in my throat. And sterilization, that hasn't changed in decades. Now, updates that are significant are cleaning validation. Oh my gosh, I am so sorry. Our cleaning validation and disinfection are the two things that have updated. Sterilization has stayed the same. There are some hiccups there, but nothing dramatical. But cleaning has the most changes, and you will see that because new standards have been published. And then disinfection also has changes for global. These are some of the standards that have been updated in the past few years for reprocessing. And if you see, a lot of them have to do with cleaning validations. And there is a huge list in there. And so if you are not familiar with them, in the last 5 years, there's been a huge push for standards for this. And therefore, we are starting to add those in for our validations as well. And they impact in your IFUs as well. And what to include in your IFUs, how to put information in your IFUs. For example, automated cleaning, right? From text to a table. So those are the things that we're starting to see changes on. Now I'm going to go through some cleaning, what changes have occurred in the cleaning validation realm. And if you think about it, if you're not aware, TIR12 that was used for cleaning validations have been updated to AAMI ST98. This is the first standard in the world that talks about reprocessing validations for cleaning. Okay, there's no other one. So if you are submitting all over the world, they are using this document as a backbone. So notified bodies in Europe are using this as well, just to make sure that you are compliant to the standards that do specify that. And some of the major changes that have happened is from TIR30 to ST98 is there were no simulated use cycles. So they have implemented the simulated use cycles now. The controls have been identified. There are more controls you have to do. Interpretation of data and what kind of analytical markers you have to test are specified. And in this chart, you will see that non-critical devices, these are devices that contact intact skin or are sitting in the operating room on the sides, right? That are non-patient contacting. We don't have to do analytical tests anymore. We just have to do visual inspection and you're fine. That's in the ISO and that's in the AAMI. So one thing we aligned with the ISO was the non-critical portion of the devices. So that is the first forward step that we have made towards that. Now, why do simulated use cycles? It was in the FDA guidance document in 2015. So we have been doing simulated use since 2015. However, no other document specified that. And if you didn't know the FDA guidance document, you wouldn't have done that test. So this standard took everything that was in the 2015 guidance document into that standard and have identified every section. It's a very easy document to read as well, but they added simulated use cycles. What this is a cycle that goes, that mimics what's happening in the hospital. When you sell your product to the hospital, what happens? They use it on the patient, they clean it, they sterilize it, goes in circles again, right? So it gets cleaned, used, cleaned, and sterilized. So we're doing that 6 times artificially. And that is how we are simulating what's happening in the healthcare. And we're doing that 6 times. Now you might say, where does 6 come from, right? We don't know. It used to, when 2015, we did 10. And then we went to 3. And now we've compromised to 6. So it will say 6 cycles in ST98. And so that's what we're following now, okay? Sample size. Sample size has increased, right? There was only 3 required. Now it's taken out. Now it's the manufacturer's responsibility to determine what sample size you're going to use, and that depends on the criticality of your product and the complexity of your product. So how complex is your product and how critical is your product? So let me give you an example. Flexible endoscopes are critical and complex, so there will be 9. All surgical devices will be 6. Non-critical devices where there's no risk will be 3, right? So that is how you would figure out your sample size. That is something that you will have to probably include in your design history file as well. The sample size you selected and why you selected it. Regulatory is looking for some sort of verbiage in it to talk about why did you use that, because it does say justify your sample size. Why did you select the sample size? They do give an example in the standard that 6 is what's commonly used for surgical devices. So what we're seeing is a lot of people are just defaulting in 6, and that's worked just fine. Regulatory is not coming back and asking why 6. But if you are going to robotics, right, robotic devices will be more complex, and it is critical. So that might need a 9. So this is where you will have to be smart about where it falls into. Controls such as negative device controls, positive sample controls, those never existed in TIR30. Now we have those controls in there for just to make sure that the analytical tests we're using are validated and they're pulling the test soil that we're using for the devices, okay? Simulated use test soils. They have focused a lot on simulated use test soils. And there's a whole section on it specifying what is a simulated use test soil and why are we focused on these test soils. So we want to make sure that we're not using just any blood soil. If your device does come in contact with blood, yeah. But what about if it's orthopedic? Do you have a bone component to it? Right? What if it's dental? Do you have tissue component or bone component to it? We just had one of our our sister facilities come to us and tell us that the dental devices we did with just coagulated blood, right? It was just a drill. They had to go back. FDA said, no, where's the bone piece of it? Because when you're drilling, the teeth is also there. You will catch on it, and there might be bone in it. Did you assess bone? No, revalidation. So this is the main piece, is the test soils you select for reusable devices have to have some sort of rationale, how it's clinically relevant. So now you have to make sure that the selection is focused on how it's used, and there is something in there. They're just not going to take straight blood and be okay with it anymore. Okay? Now, so that is the first step. The next step is soiling methods. How is this device soiled, right? No more immersion, spray, whatever you want to do, direct inoculate. All that is out the window. Now you have to simulate it, right? You have to see, does it get handled during surgery? Let's handle it. What portion is immersed into the body? What's going to immerse in there? What portion will be sprayed? Oh, the cable might get sprayed. Let's focus on that, right? So the method of soiling is as important as the selection of the test soil. So the document specifies a lot on the method as well. Now, if you see here, I've got a demonstration. Now you'll see that the devices, there is one handling with soil, and then you see that we have the entire scissors covered. This is worst case testing, right? They're focused on worst case testing. What if this happens? Did you assess that, right? And if you didn't, then that is something that they'll challenge you on and ask you about. So very important is that you want to make sure that when you are also validating these, they are mimicking the procedures that is being followed in the hospital or during surgery, and you take a notch up and make it worst case as well, right? So we're also making it worst case. This is another example. Robotic devices. These are cauterizing devices, right? We need the entire equipment. Those big machines that we have to validate, we have to program, and we have to get a kidney submerged in blood and use the device for simulated use. Okay, so that is another one. So now the regulatory is asking for, we want to see real use in the lab setting. No more small, just focus blood, oh, it came off, we're good to go. They want to see the real processes and how it's being worked. And then we have all this, and now we have another challenge. They have to be dry to the touch. They have to be dry to challenge the cleaning process. So we soil them heavily, and then we have to clean them after it's dry. The reason for that is if you think about it, when devices don't get cleaned right away, and I have a picture down here that does talk about, but does show you that there is cases lied there. And you will see that there's tons of cases there. And if you look at the cases, you might be like, okay, this is it. Think about 4 of those tables that have to be cleaned in 8 hours of the shift. Are they looking at your IFU? No, they don't have time for that. Are they processing according to how I validated? No, that's not happening. They have their own procedures in the hospital, right? They have their own detergents in the hospital. What I validate with is not what they're using. They're going to buy what's good for them, right? They're going through detergent like crazy. So think about it. Cost efficiency for the hospital. They're going to buy drums of detergent that's cheaper and that can be used in small doses, right? So if you have a validation and you did it in the middle dose, what if they're using half an ounce and we validated 1 ounce, right? So when the instructions for use say prepare per label claims of the detergent, they're going to go, if the detergent has a dose of 1/4 to 1 ounce, if I'm a hospital, what am I going to do? I'm going to do a quarter because I'm not going through those detergent fast. And I have to change these drums every week. I can't afford it. So that's what I'm going to do. So our validation will also do that, right? We'll go to the quarter ounce and do it that way as well. But speaking to this, we have to dry it. When I started 20 years ago in this industry, in reuse industry, we did what the hospitals did. We took a surgical towel, wet it with water, and placed it on the devices to keep it moist. So when it goes down, it's still wet. That was the normal procedure. Everything went through the regulatory fine, and then it moved to, they're not putting this on there. We don't have water in the surgical unit, so we're not doing this. So that got thrown out the window, right? And so they're like, okay, what now? No, we just leave it there. Okay, no, some hospitals are like, no, we do spray on them, so stuff like that. But I remember I was in an audience like this, and John Hopkins was speaking. And one thing they said was, we process 14,000 instruments a day. Do you think we're looking at your IFU with those 14,000 IFUs that are coming across? Please align the IFUs. Make them so if you have forceps, it's the same IFU from company A, B, C, D, E, right? Things like that. So they brought attention to manufacturers like, why do we have 15 different IFUs for the same product? And hospitals get pushed on that, right? They're like, well, which one do we pick? We can't do different for each one. And so things like that. But that got stuck in my head forever. And I thought about it and I was like, yeah, they're right. They're right. In a high-speed environment, reusable devices, you have to turn them around very fast. And you can't be looking at every single IFU, reading them, and then processing them. So they have their own internal processes they go through, that they make sure there's a safety factor associated with it. What I mean by that is most of the hospitals have incorporate, you clean, you place it in an ultrasonic unit, whether your device can take it or not, go in the washer. And so they've put plans in there just to make sure that they have assessed for safety. So when you're selling a product, you just want to make sure that you understand that is happening. But we challenge it with dry to the touch. So it has to be dry before we process. So we use worst case test soils, worst case contamination, and we dry them, okay? 3 variables already worst case. And then we do the automated cleaning cycle, right? So for automated cleaning cycles, we have a cycle parameters. I'm sure you've seen these cycles. And I've outlined thermal cycle here because that is not part of the cleaning. It happens in the hospital, but for regulations, and it is in your IFU, but we're not going to use that. We're going to omit the IFU out and validate the rest of it, because IFU is now a disinfection step. You just added a disinfection step to the cleaning validation. And you have to assess those separately. Disinfection separate, cleaning separate, because the endpoints are different. And so they are assessed separately. And so for simulated use cycles, like we talked about, those are included because that is happening in the hospital. But in real life, in validation world, we have to break them apart and validate them accordingly. Now this is a table that every regulator wants to see. So if you don't have it in your design history file or you don't have it, I would highly recommend that you have this table. It is very easy for FTA or notified body or whoever to look at it and be like, okay, you guys challenge the process. Otherwise, you have to sit there and explain the whole thing, pull up the protocols, look at the protocols, and all of that stuff. This shows what your IFU is and what the testing lab did, right? It says per label claims, we take it to the lowest. It says soak for 2 to 5 minutes, we take it to the lowest. We'll do 2 minutes. We're not going to do 5 and we're not going to do 3. We're going to do 2 minutes. And things like that. Processing steps that will impact the end point are the steps that we will reduce. Now, if you reduce everything, you're going to fail. So we have to be very smart on what parts of the IFU we are reducing. Detergents, right? Are detergents what I am using are being used in the hospital? Right? No, they're not. So you might be like, then what's the point of this test? Why are we doing these validations? Because every country will have their own detergent. And every city will have their own detergent. They're not the same. Hospital A, B, C have their own detergents. And I have my own that I validate in the lab, right? So your IFU will say, use an enzymatic neutral pH detergent, and that should qualify the rest of them, right? We're finding that when you say that, there are different enzymes in these enzymatic detergents. So the ones that I use only has one enzyme. So it's more challenging. Hospitals use maybe multi-enzymes, but some might not. So if you're testing at a lab, you want to make sure that you're challenging your at least the detergent that they're using doesn't have like a multi-enzyme, because now you are not doing service to what's happening in real life. Okay? And one thing to remember about detergents is Europe they use alkaline detergents, right? US uses neutral pH detergents. So now we're looking at two different global perspective of how these validations should be performed. So when you do neutral pH here, it's fine, but you probably will get calls from the hospital, which we've heard from manufacturers asking, can we use alkaline on your product? And you don't know that. Will it clean? We don't know that. So we do see often manufacturers validate using both detergents just to have that in their design history file, that we have addressed alkaline and we have addressed, and they are equivalent. The results are both fine. But we do see that often change, okay? Now rinsing. New standard just got published in August, AAMI ST108. This is water quality for reprocessing. It is for healthcare users, it's not for manufacturers, but it is good to know that this standard is updated. It is for reusable devices, and it is on the healthcare. They are now mandated with this standard to do monitoring programs, okay? They have to monitor their water. They have to monitor all of their steam, sterilization. Water systems also have to be, because if the water is not clean for steam, you will fail. It does impact sterility. And same with disinfection, same with cleaning. If you have hard water, what happens to the enzymes? They inactivate them. They don't work anymore. Now, if you think about waters all over the world, I mean, let's talk about waters in the United States, right? We know that there are certain certain states that their water quality is very bad. And hospitals don't have the infrastructure to have the piping to make those equivalent to what they need to be, right? So this is a huge impact for hospitals, but when you see wear and tear on your products, it is water-based majority of the time. Water is one of those things that are hidden in the background that you think, oh, it's just water, but it can damage very hard. And I'll show you pictures in the next slide. That's water. That's water damages. And in this standard, they have there's a whole annex of just pictures that are specified for damages with water. And this is from my washer. These are my dunnage devices that I've used, processed over and over again with medical devices that I'm validating, but these are dunnage just sitting in my dunnage. They've been probably a couple of years, but this is what they look like, and half of them have even fallen apart. And these are medical grade stainless steel, right? So having proper water quality mentioned in your IFU is a huge factor, right? We will do it, but you also want to make sure that you write when to use the critical water. It needs to be in the final rinse. Your cleaning validation will be done with tap water, but now monitoring is required, so they will have to figure out how to do the monitoring of this tap water, but you want to make sure that they are in compliance of that as well, right? Now, these are cleaning validation acceptance criterias, and you will see that ST98 has one, ISO 15883-5 has one. Now, keep in mind, FDA does not recognize the 15883 standards, the whole series. So this is for Europe. Okay, so they have action and alert levels. What they mean, we don't know. ISO doesn't specify it. That is something that we need to interpret. But what I'm starting to see is alert level is now their action level, right? So they're more stringent on what these validations should be than what FDA is, right? For example, protein is 6.4. Germany, Switzerland, France looking for 3 micrograms per centimeter squared, right? Half of what's required by US. So when you do validations, make sure you take that into consideration. Otherwise, you'll have to do 2 validations. So keep it lower end. And if you hit the lower end, great. You met both the criterias. So make sure that when you're designing your validation plans, that is on there. One thing to remember, they have ATP on here, but ATP is not used for validations and should never be used for validations. This is only used for healthcare to monitor your products, right? When they're reprocessing, they have to do some sort of monitoring, and that's what they use ATP for. Is it got cleaned, it didn't get cleaned, and that's what we're looking for. The good part in the standards is non-critical devices. You just have to do visual inspection. And so that is, I soiled it with test soil, I wiped it, and there's no blood on it, good to go. And that's all you need for cleaning validation for non-critical devices. And these devices are devices that come in contact with intact skin and devices that are sitting in the OR in the outskirts. Okay, there's a computer, touchscreen, keyboards, ultrasound consoles, stuff like that. Now here is some of the different criteria for protein in different countries, right? So you also want to be aware of that. Some notified bodies are asking for 10 micrograms per instrument, right? No matter what you did. So now you have to reevaluate that. So be careful when you do cleaning validations that you are looking at different regulations. And this is the only one that has different regulations in different countries. Okay, majority of disinfection and sterilization is the same, but cleaning is where you will see the differences. Now, this is in the standard, and we have talked extensively with the FDA on it, but they haven't mandated yet where you would want to where they're looking at your results and a standard deviation is applied to the highest value of your analytical test that you do. But it is coming. It's in the standard for a reason. But I think they're giving a grace period before they start to do that. So from here on, any validations that you do, you want to look at your, where's the, because if you see 0.9, 1.1, 4, that should already alert you that there is something happening. Why aren't they similar? This is a made up numbers that we add. This is straight out of the standard. So we made these numbers up. But if you ever see numbers like this is where you question what's happening, okay? Because there's something wrong. And, but if you see the standard deviation of those 6 data points is 2, it's added to 5.6 and you get 7.6, it's a failure now. So you would have passed earlier before the standard came out. Now you would fail. And when you fail, the standard says you have to reassess with a larger sample size. Okay? What is that larger sample? If you think about standard deviations, how much samples do you have to add to even minimize? So you can add 100 samples and you might not bring it back down because the original results have to be included, right? So that brings you to testing to compliance at that point. So we're working with FDA to figure out a game plan for this. And I think that's why they haven't enforced it yet. But once the game plan's on, I'm pretty sure that they're going to start asking you about it. One thing that I want to alert, I just talked about 2 hours ago for design history file, is look at your master products and make sure that they're within. Okay? Because if they're not, that is something that will be red flagged. So they're within that 6.4 after you do the standard deviation. Because if they're not, that is going to be a red flag to just look at it. We talked about non-critical devices, the standard, ISO, and also specify that you just have to do visual inspection. So that should be sufficient enough. And so it's just not one standard. It's universal. Even everywhere in the world that has been adopted. So that's a plus for us. Now I'm going to go into disinfection validations. AAMI TIR12, which is a technical information report, got published in 2020. And in there, there's an extensive information, a section on disinfection. We don't have a lot in disinfection for reuse. So we added that into this section. And we'll probably, I'm taking it further, and I'm going to write a standard on it just to make sure that manufacturers have an idea of how to select what categories are going to be for disinfection. But ISO 17664-2 also got published, and that is for non-critical devices. So if you have non-critical devices, this is the ISO standard that you want to look at as well. Now for disinfection levels, we have critical devices that need sterilization. There's no disinfection for that, okay? If you have a critical device and you're saying, well, we have to figure out a way to disinfect them, that's not going to work with regulations. You will have to do sterilization. If you can't do steam, it's going to be low temperature sterilization modalities, VHP. EO is not for reusable because it's a fast-paced environment. We have to wait for residuals. It doesn't work that well for us. But VHP is what is the next standard next to it. Semi-critical devices is the devices that contact mucous membranes. So those should be sterilized, but however, if you can sterilize them, you can high-level disinfect them. And these are like ENT devices, okay? Bronchoscopes that are going down, ears, eyes, things like that. Vaginal probes, that's what we're talking about for semi-critical. And then non-critical, we just talked about, those are the ones that are intact skin or just in the healthcare facility, helping assist critical procedures. Now, if you look at the acceptance criteria for all of these, look at this non-critical acceptance criteria. It's 6 log reduction of 4 organisms. You're not going to get that with label claims, okay? Your instructions for use should never say follow label claims. Okay, that needs to be validated. And we have never, ever validated something on label claims. Why? Label claims for disinfectants are done on coupons, not on medical devices that have all these different design features that are difficult, all those crevices that I'm inoculating with bacteria, and now I'm trying to wipe them off. All the materials, most of these devices are silicon-based, right? They have different textured surfaces, stuff like that is not used for these chemicals. So you will never meet the instructions for use on label claims ever. So we always increase that. And that was something that when we trained FDA in August, that blew their mind because they're like, you're going against EPA regulations. Okay, we've been doing this for decades. So EPA registration is for the disinfectant claim. It's not for reuse devices, okay? It's for hard surfaces. We're using those on medical devices now. So we have to evaluate them separately. But that is something that I want to bring to your attention. A lot of people will say, follow per label claims on the instruction. we're never going to meet that, and that doesn't happen. However, hospital still does it, whether you have it or not, right? They're going to just wipe and move on. But for the validation, we will say wipe for 5 minutes. Nobody does wipe for 5 minutes. They do it literally a second, and that's the reality of it. Validations are a little different than what happens in the real world. Now with the intermediate and low level TIR12, it's a gray zone. It's non-critical, but where do you put it? It depends on how your device is in contact with, how close it is in contact with the patient, or how should we evaluate cross-contamination. That's what manufacturers are. There is no list that says if you're here, you do this. If you're here, you do this. These two are both in the non-critical zone. You will have to evaluate if it's a intermediate level disinfection or low level disinfection. And that is determined upon the proximity of that device to the body and if it's cross-contamination. If it is touching the intact skin, intermediate level. The only problem is the ones that are sitting in the OR room. How close is it to the critical procedure will determine the next phase. And we talked about that. And then there's interim thermal processing. This is the thermal that's required by EU. So if you don't have a thermal disinfection step, if you remember, we talked about that in the automated cleaning. I highlighted with orange. This is where you have to validate for user perspective. Nothing to do with your device. This is only for user perspective. For automated washer, you put it into the washing machine and then it opens in a clean side. They're using bare hands to remove it. So this is only for user perspective, but it's now a requirement that you have a thermal disinfection if you're going to submit in EU. Your MDR will ask for that. Okay, so if you don't have it, you probably want to assess that. Sterilization, the IFU hasn't been impacted. Okay, it is the only thing that you will see if you haven't done yet is that dry time testing has to be done separately. It's no longer part of the sterilization process. It's written as one, but they're split up. The sterility assurance level, which is the SAL testing with the bacteria, will be separate from dry time testing. And the cycles are also different for those. So you have to have two different testing for sterilization. And I'm only including steam here because 80% of all reusable devices are done through steam. It's a fast process. VHP takes too long. EO has residuals. And it doesn't work well for other things. So if steam is what is the most market for reuse. These are some of the recent changes that are going to happen for standards that you probably want to look at for your instructions for use as well. I won't go through the changes because it's written up here, but we have a new VHP document, and we don't have dry times anymore. They used to write dry times in the table. They've taken it out. So now you have to determine what the dry time for your product is. So if you have a big case, the highest we've seen is 70 minutes. Don't go past 70 minutes. Hospitals are not going to do past 70 minutes. So if you can't make your dry time in 70 minutes, that means reconfiguration of the tray, or you are placing your instruments in a way that steam can penetrate easily in and out, okay? Lumens facing the walls, things like that. End of life validations. This is a new thing. Regulatory is asking about have you assessed your end-of-life use testing? It's not a requirement test, but EU and MDR are asking for it, right? Have you at least assessed it? And do you have data? How long can your devices be used? So now we're crossing that area where before it was not required. You can just write in your instructions for use. Now they're asking for data for it. And if you see this picture here, I'm sure you guys have probably seen it in the news. It was a while ago, but it was a study done by Michigan Health, and it was only used 20 times. So if you see the design was not meant for reusability. The design was meant to throw away. You can flush in there. If it's a suction catheter, you can't brush. there is no way a brush is going to go in there. So you flush, but everything else gets stuck there. And then you sterilize it. Sterilization doesn't do anything if you have blood in there or tissue in there. It's just something to make you feel better. Doesn't do nothing. So if you don't clean it out, you're not sterilizing anything. So that is what brought up end-of-life use testing in the eyes of regulations and in the eyes of the industry right now. We already talked about that. Now, one thing you want to remember about end-of-life use testing is ISO 10993 already had this language in the document. It hasn't been reinforced, but is being reinforced now because they know about this, right? And we're educating manufacturers that this is coming your way. Because it says biological safety shall be evaluated, and in the update, they're updating it again, and they are making sure that this is going to be addressed in there, okay, for reusable. And you will have to probably do some sort of assessment, a risk assessment or a toxicological assessment. Something will have to happen if you're not doing the testing, but you just cannot not address it, right? The ISO and FDA guidance does write that as long as in your IFU you specify to the user when the devices can no longer be used, you're fine. But you have to assess that. And how do you know that if you haven't tested anything? Right? And when you do end-of-use life testing, it's not the engineering functionality testing you guys did that's being done. It is soiling, cleaning, and sterilization. It's what is your device being sold to the hospital for? That is what you're going to get it through. And that is how it's going to show, because they will ask, okay, so you've written this, how did you evaluate that your device will discolor here? How did you evaluate that this will happen? Those questions will arise and come, so you want to be prepared for that. MDR says the same thing, but we're seeing notified bodies from EU asking for end-of-life use testing. So there is a push for that. So you probably want to evaluate it. There are many avenues that you can use, but we do have a whole team that can help you with that as well. Now the UDI number, okay, our 1R got extended to 2028, so you have time, but the UDI number has to be on the device, okay, because that is how we're going to process how many times it can be reused. Will we be ready for it? No, not US, but will EU be ready for it? I'm pretty sure they will be ready for it. And I don't know how many people have started work on the UDI, but hopefully it is on the works because they've extended it several times. So it may be extended, I don't know. But they've extended to 2028. But that is something that needs to be on the device to make sure that it can reprocess as many times as you specify. And that's what the ISO 10993-1 is going to start saying for biological evaluations. These are some of the standards that are for reusable devices. And if you have any questions for me, I can take it now. I do have some slide rulers in the back for reusable. These are slide rulers that tell you what tests are requirement for a critical, blood contacting devices, all different cardiac devices, and you just have to, it will tell you what's required, what's recommended. So I have some slide rulers in the back. And if you want to talk to me later or learn more about other tests, we are in booth 3321. Thank you.